Emma is 8 years old. She survived a bone marrow transplant for leukemia. Now, she's on an antifungal for an infection that kills over half of the patients who get it. Her doctors prescribe the standard dose. But here is a terrifying truth. No one knows where her levels land because this drug behaves differently in every human body. Too little, the infection wins. Too much, organs fail. Because the safe range for this drug is small, monitoring is mandatory. Up to 12 blood tests per treatment course. But that means dragging Emma back to the hospital month after month while she is fighting for her life. There is no commercially approved microsampling product for this antifungal in the UK today. Capilla changes that. We developed a finger prick device that captures a small drop of blood from the patient's home and sends it to existing NHS laboratories. No clinic, no veins. Our device has less than 2% variability. The leading alternative, as much as 20%. In the UK, there are 3,000 invasive fungal cases every year. That means 90,000 monitoring tests unserved by Capilla. We are seeking £500,000 to validate antifungals on our platform and run our first NHS pilot study. Emma doesn't need a revolution in medicine. She needs to monitor her levels from home at the right dose every time. Thank you.
Do you have an idea of currently what's the size of that market? So what is the annual cost of that testing in the UK?
So in the UK in particular, invasive fungal cases. So we can look at two buckets of patients, for example. So bucket number one, we have the invasive fungal cases. So there's latest clinical studies from 2018 that show there are around 3,000 invasive fungal cases every year. And that's in 2018. And we know now that it's almost certainly have grown. So that's around 3,000. But we look at the second bucket of patients. We're talking, for example, the prophylactic, the preventative, so the high-risk stem cell transplant. They add onto our patient pool of population, and that adds up to totally around 9,000 per year. But because those patients are on monitored for drugs, times that by 10, we're talking around 90,000. Now, to be transparent with you, that isn't venture scale. It is more of a clinical proof of package, but our value for our platform is not the drug itself. It's going to be a multiple drug platform. So we are looking, for example, at targeting not just antifungal, but also antibiotics and immunosuppressants in particular.
Right. So what is the biggest source of variability compared to venous sampling? And is this a device that you're going to be marketing?
So the biggest failure in venous sampling is not necessarily the accuracy. I think the reason why we have built this device is because we're putting less emphasis on the patient experience. So as I said, our device, we want to be able to take a finger prick sample. So that finger prick, not only is it less painful or less invasive compared to a venous sampling, it also is more convenient for the patients because they can send that sample via post to the NHS lab. And this is something that's already done with the NHS currently with, for example, with bowel cancer screening programs. And as well as that, having done some surveys with the patients in particular, they have mentioned that not only is it convenient for them, but it also restores dignity and maintains their confidence. And in terms of, what was your second question? Sorry. Yes, so this is an in vitro diagnostic device class A. And the beauty in the class A is it's self-certified. So it's a sample collection tool, not a diagnostic. The diagnostic always happens in the NHS laboratory lab. So with the Class A, there's no notified bodies, there's no conformity assessments.
You still need to do a proper clinical trial to get adoption from clinicians, though.
Mhm.
So have you costed that?
So with our in vitro diagnostic device Class A, we actually, with the funding raise of £500,000, we're not necessarily doing a powered clinical study yet. We're doing a NHS pilot study. But for our device, in vitro diagnostic, it is a performance study. So we're doing a paired concordance study to compare our device with the standard pathway, which is the venous bloodline. So with the pilot study, we're looking at around 20 to 30 patients, ensuring it matches, not only does it match the venous bloodline, but it also, in some ways, exceeds it, as I said, with the 2% variability.